All right, everyone, welcome back. Let's go ahead and get started with the next session, which will be chaired by Anne Hermanstad. Anne.
Great. Thanks, Janine. So let's start things off with Ned Wingreen from Princeton.
Thanks very much. And thanks to the organizers for organizing this very interesting forward-looking meeting and giving me a chance to talk here. Let's see. I hope you can see everything.
It looks good.
Great. Looking good?
Yep.
Okay. Great. So yeah, my title today is Gonna Make This Garden Grow, Learning from Algae How to Concentrate CO2. So let me give you a brief outline of what I'm planning to say. So I have been involved in a project with Martin Jonicus, who's a faculty member at Princeton, and he has a long-term interest in carbon concentration mechanisms. These are important for various organisms that do photosynthesis in order to make them more efficient. But it turns out that not all organisms take advantage of these mechanisms. And sort of his long-term goal is to teach crops to do the things that algae and some other plants are already able to do. So this is part of a theory experiment collaboration that's ongoing. And our part of it has been to develop a quantitative model for the carbon concentration mechanism in algae. And so I'll tell you a little bit about that. Time is short, and I'm not suggesting that, that this is the entire future of 4D cell physiology, but it's an example and maybe one that we can draw some lessons from. So I'll teach you what we learned from modeling, and then try to generalize a little bit based on this and my other experiences in joint experiment theory collaborations to give general thoughts that I hope will be relevant to this initiative in 4D cellular physiology. So our story starts with this enzyme Rubisco, and Rubisco is great. It is actually the key enzyme for fixing CO2, but it's got a problem, and that is that it's very slow. So Rubisco, only turns over a few molecules of CO2 every second into biomass, and this can really be limiting for the growth of plants and other photosynthetic organisms. So in particular, is limiting for things that we like rice and wheat and other crops. So wouldn't it be great if Rubisco could be made more efficient by having high levels of CO2 right where the Rubisco is, make each Rubisco not faster, but just have more of its substrate around so that it's in a bound complex and therefore is producing more fixed carbon per unit time. So there are, in fact, organisms that do this. So algae do this, cyanobacteria do this, some C4 plants, and you can see how big they are, manage to do this. And I should say the thanks to Martin Jonicus is not just for these slides, which I've shamelessly stolen from him, but also from inspiring our interest in this system, because there's a lot that's not understood about how this works, and understanding it would actually be helpful to his goal of engineering these systems into our crops. So, going on from here, you have plants, and there's a chloroplast, and that's where the photosynthesis is happening. But the Rubisco is spread out through the chloroplast, whereas in algae, like Chlamydomonas, which is our favorite organism of choice, the Rubisco is clustered together into an organelle called the pyrenoid. And it's there that the CO2 is concentrated by this carbon concentration mechanism. So there's the pyrenoid, there's where the CO2 is. And if you want to see this, in an organism, here's an electron micrograph. Of course, there's lots going on because this is a living cell of an algae, and it does all sorts of things. It swims around, it phototaxes, and so on. But here you'll see that there, right in the middle of this chloroplast, is the pyrenoid, It's surrounded by these starch platelets, which are a place of storing carbon during the day when it's doing photosynthesis that it can use at night when there's no photosynthesis going on. And also you can see these little purple things, which are extensions of the thylakoid membranes where photosynthesis is happening into the pyrenoid. And this is really important for the mechanism by which this organism concentrates CO2. So what's supposedly going on in these organisms is that by virtue of this efficiency, they are able to assimilate large amounts of CO2. And in fact, something like 35% of the global CO2 assimilation takes place in pyrenoids. Organelle I had never heard of until now, but now, just like Martin, I've come to love the pyrenoid. And of course, we should all love it because without it, we would have much less fixed carbon. Things would be even worse than they are now in terms of global warming. So, so how is this supposed to work? Well, here's a blow up of an electron micrograph, and you can see here's the rubiscos, which are kind of large enzymes. You can actually really make them out. They're jam-packed into this organelle, but room is made for these tubules. And what's supposed to happen is somehow CO2 is supposed to actually get pumped into these tubules in a kind of carbon circulatory system and released into the pyrenoid where it can be processed. And so the question is, well, how is this supposed to work? That looks rather miraculous, that this is supposed to be a circulatory system. And here's the idea. And the idea is that outside of the cell and outside of the chloroplast, there is inorganic carbon. You can't make carbon from nothing, but it's in the form of CO2 or bicarb. And that's going to enter into the chloroplast where the pH is different inside than outside. And therefore, you can have enzymes, carbonic anhydrases, which can convert CO2 into bicarb, preferentially at pH 8. But then these somehow have to get into the lumen of the thylakoids. And from there, they have to get into the pyrenoid where they can be released as CO2 to be processed by Rubisco. And so, you know, this is a nice picture, but the question is this really going to work? And to address this question, we set out to do some modeling, and we developed a reaction diffusion model for the CCM, which required lots of choices of how we were going to do this. But in the end, we ended up with a spherically symmetric cow model. But the cow, inside its spherical symmetry, it has a lot of subcompartments. And so there's the stroma of the chloroplast, there's the matrix of the pyrenoid where the Rubisco is. Then there are these tubules, which we model, even though it's a 3D model, as compartments, which traverse from the chloroplast into the Rubisco. And so there's quite a bit going on there. We solve that and we find that it doesn't work. And so in fact, this diagram shows what happens. Inorganic carbon comes in, everything's going great. It gets into the tubules, it goes from the tubules into the matrix here where there's more carbonic anhydrases, which turn the bicarb back into CO2. And then only a tiny bit of it gets fixed before it diffuses right back out of the pyrenoid. And so this was a good lesson that, you know, you got to put real numbers on things. And what we thought then was, well, okay, we're not including everything in this. What about that? This feudal cycle is what's the problem. It's just going to go around and around without getting processed. But what about the starch sheath around the pyrenoid? Maybe if this is a diffusion barrier to CO2, that's going to help. And indeed, with that included in the model, everything now works. Everything works as it was before. And now the CO2 is trapped within the pyrenoid by this starch sheath and doesn't let the CO2 escape. So that's good. And of course, there's lots of other things going on. We have to put the carbonic anhydrases in the right place, or you get other feudal cycles. But I just want to sort of cut to the end and say that we get no more feudal cycle, and we learn a few things about how we might engineer this into crops. And sort of the important thing is to say it's feasible. This system was based entirely on pH differences among compartments. And it wasn't immediately obvious that was going to work, or what were the essential components. We find that indeed you can do this without necessarily adding pumps to a system, which simplifies the engineering problem tremendously. It's energetically efficient. Surprising to me, only 1.5 ATP per CO2 that gets pumped in and fixed, which seems quite cheap by the scale of other energy costs. And that you've got to avoid these feudal cycles. That's another lesson. So where does that leave us? Challenges ahead. Well, there's lots of things we don't understand. We have snapshots of how these things look, but we don't know how they form. There are these tubules. It turns out there are mini tubules inside the tubules. We don't know how they form or what they're doing. We don't know how they get to be one pyrenoid, how it gets positioned inside the cell. We don't know how the transporters or the anhydrases are localized, and we don't know their rates. And we don't really know how to engineer it into crops, but theory does help. It tells us of all the things you could do, what's the right order of events in an engineering problem. But I want to highlight that these challenges are not just for the CCM, they're really challenges for cellular physiology more broadly. So this question of how things form, well, that's development and self-organization, where there's still a tremendous amount to learn at the cellular level, the physiological level of how the structures that are going to be important for physiology actually develop and self-organize and are maintained. And then this question of parameters. This is a huge problem. There's all of these different components, and they all have different parameters that also depend on things like temperature and pH and salinity, possibly of cofactors and so on. And it's really hard to measure these appropriately in vivo to know how things really work. And finally, this question of physiology, I mean, one of the long-term goals is things like tissue engineering. And so again, we're going to be faced with these engineering applications. And I think theory can really help us in all of these, admittedly in partnership with experiment. So my general lessons in the last minute, what can theory do? Well, this is probably trivial for this audience, but theory can provide a systems level view. It's hard to see the forest for the trees when you're actually doing real experiments, but we theorists can sit back. We don't have any cells to keep alive or equipment to buy. So we can help you with a systems level view. Maybe more interesting than that is we can think about what would constitute optimality. What's the best the system can do? And if it's not performing near optimality, what are we missing? What are the constraints that we don't know about? And it can help us test ideas. This is a great idea for how the system can work, but what happens if you put real numbers on it? And finally, the question of closing the loop is how to best build and maintain theory experiment collaborations. So that may be something for the discussion at the end. With that, I'll end. I'll thank the team, Chen-Yi, Alexandra, Neil, and Martin. You can read about this more, and thanks to our funding. And so I will take any questions.
Great, thanks so much, Ned. It looks like Daniel has a question. Daniel, if you want to unmute yourself and ask your question.
Okay, just a very quick addendum to a comment which I think slipped by rather fast, which was the stage at which Ned said, well, this turns out to be wrong, it doesn't work. And that role of theory in being wrong, so one can sometimes rule out whole general classes of possible ideas and things, is a very important role. So I just wanted to add emphasis on that.
Thank you. Hashtag things can be wrong in theory.
We should definitely come back to that in the discussion. I think that would be a great thing to discuss together. We also have a more technical question in the Q&A. So this person would like to know if you've tried to use carbonic anhydrase inhibitor like acetazolamide, and how does that affect the question that you're trying to answer?
Well, I mean, we as theorists, we can do anything we want. And in fact, we have done things where we've considered lots and lots of the known mutants and how they perform. For example, there's a very interesting phenotype that dies in air level CO2, but actually recovers at even lower CO2. From an experimental point of view, certainly the system relies on these carbonic anhydrases. If you really inhibit them, this is not going to work. But there are more subtle issues there about where they have to be and how well they have to function that, again, we can address theoretically as a guide to experiment. So I think that's an important role we can play.
And then I think we have time for one more question to squeeze in. So from Janne, has anyone been successful in building an organelle like the one you described for concentrating carbon, and how big of an engineering challenge is that? And also, if you might be able to unshare your screen so that we can all.
I can definitely do that. So this is really Martin Jonicus's long-term goal, which is to engineer a pyrenoid into organisms that don't have one. And this is a project that's not involved in labs around the world. So there's real progress. I mean, they are able to, reconstitute parts of this, but still early days in getting a full pyrenoid to be reconstituted. And get the rubisco to condense carbonic anhydrases into the right place. So I think these are, again, general challenges that are going to be faced as one thinks about the engineering aspects of cellular physiology.
Great, thanks, Ned. I think we should move on to Hyun Yuk, who is our next speaker from University of Massachusetts Medical School.
All right, let's see. Can you see my pointer actually on the screen?
Yes, yes, we
okay.
can.
All right. So I'll just start. So first of all, I want to thank the organizers for inviting me and also organizing this workshop. I'm an experimentalist at the University of Massachusetts Medical School in the Systems Biology Department. My lab studies microbes and mammalian cells that are either dormant or growing incredibly slowly. And our goal is to understand basically why cells, what it really means to die very rigorously, and to know whether life can stop indefinitely and resume again. And if that's possible, how you can do that, and if not, why that's impossible. So that's sort of what my lab does. And today what I want to do is tell you from an experimentalist's point of view, tell you about sort of my experience of engaging in this theory model back and forth. I'll just give you some example from my lab and just my own experience in working in this, working with this sort of back and forth. And I should say I'm not a deep thinker, and I don't think I have any deep insights into this, but I'll just tell you what I experienced. And in particular, what I want to focus on is actually where to start this feedback, because you do have to start somewhere. And to put it simply, you can either start with a model, which means that you kind of propose, you do some calculations from using a model or theoretical framework that you have, you make some predictions, and then you do experiments to test those predictions. Another way to start the feedback loop is to start from experiment. And that is really what I'm really going to talk about today. And that's what my lab has mainly been doing. And what that really means is that in the case of my lab, we tinker around with some cells in the lab, and we discover a phenomenon, usually by accident. And then we either build a model to explain the phenomenon or do what I call sanity check. And that means that basically you understand the zeroth order mechanism that explains the phenomena, but the mechanisms involve rates of different things. So you want to put these into an equation and see if these equations produce the dynamics at the right time scale that you actually do observe in an experiment. And something that really I wanted to maybe discuss today and share my thoughts on is really, how do you know where you start, right? And for me, I think it really comes down to knowing whether the assumptions in the model, so that is the starting ingredients of your model, are true or false. And very often, almost anything that you can think of, it's not quite clear whether the assumptions are really true or only true in some cases, or and if so, when exactly those are true. And that's I think something I want to focus on today. And I'll give you an example from my own lab where we thought the assumptions were really true. I mean, that's what we have learned and have seen in published papers. But it turned out that very simple experiments show that the very basic assumption was false. So in this case, had we started out with a model with these assumptions, we would have had, garbage in, garbage out scenario. Okay. The other thing I want to mention is that it's also not quite clear whether, it's not like you have assumptions and you build a model and you have some predictions and you test it, and then you can tell whether it's true or false. In some cases, that's not really clear, and that's something I will mention as well. Just to be very concrete, here's an example from a colleague of mine, Rob Brewster here in the department, who does a very, especially great job of taking this approach where you start out, in this case, with theory, a theoretical framework like equilibrium StatMac to understand gene regulation in E. coli. The details aren't important, but basically Boltzmann factors dictate how many transcription factors are bound to a promoter. Separation of timescales lets you use that framework, and you predict it, and you do experiments in E. coli, and all these data points match the theory in the line. So that's really one approach, and I think many of us know that this is the kind of approach that was very eloquently and in a very fun way described by Rob Phillips in this review article. And there's one sentence I want to highlight here, and he says here that figure 1 theory, which means that you start out with a model as figure 1 in a paper, is about living dangerously. And I think what that really means is that it's not dangerous to accept statistical mechanics as being true, of course, but here you have to accept that all the binding energies, which you cannot really measure in vivo, are true, and so on and so forth. In other words, it's very dangerous to it's risky to kind of accept certain assumptions in a model as being true, right? And I think that's how I think of it. Now, of course, if you publish a paper where figure 1 begins with a model, it's not so dangerous after all, because you have a paper, but to get there, it's really risky. Now, from my own lab, I want to give an example where we started out with an experimental finding that showed that a very basic assumption that we had about a system was wrong. I won't go into all the details, but basically here what we were interested in was trying to study how yeast cells, Baker's yeast cells, divide and combat heat shocks at high temperatures. And the very basic view that conventional view that we had, that was certainly my view, was that yeast cells autonomously combat heat shocks. We think of proteins unfolding at different high temperatures and so on. And the main idea is that at sufficiently high temperatures, cells fail to replicate, right? And the way to represent this phenomenon is something that we've all seen in papers and textbooks, which we reproduced here with a plate reader. So basically, in the case of yeast, as you hit 40 degrees Celsius, the population stops to grow. In this case, what we did was we did some very basic experiments which showed that this picture was not really correct. And this graph that you see here, which evidently is true because we reproduced it, is actually an illusion coming from the fact that one other variable, which was not thought to be relevant, but that variable has to be varied in order to see the full picture. And the basic phenomenon is that yeast cells help each other combat heat-induced damages at high temperatures. The way that happens is that basically, if you do a simple growth experiment at this supposedly habitable temperature below 40 degrees Celsius, if you start out with a macroscopic but still a low number of cells in a population, that population will never grow and will become extinct. But if you have a slightly higher number of cells to begin with, then the population will grow. And experimentally, by tuning now the number of cells you start out with and the temperature at which they are incubated, you can determine this phase diagram in quotes. And so this is an example where we didn't use any model to produce, to observe this. And it's an example where the assumption, which was that cells autonomously grow at high temperatures and combat heat-induced damages, turns out to be wrong. The actual biological mechanism turns out to be that at sufficiently high temperatures, yeast cells secrete an antioxidant, glutathione, which cleanses the environment of reactive oxygen species that get created from non-carbon, actually, nutrients. So in this example, we built a model with very simple parameters. And long story short, it is able to reproduce the phase diagram that you see here, and it uses one fit parameter. So, for me, what I like to think about was, so this took a few years to do. Diederik, the grad student, really an excellent grad student, worked really, really hard. But at the end, we're left with a model that applied to this particular phenomenon, but could it actually apply elsewhere? And that's something that I like to think about. And in this particular case, it turned out that the actual model does apply to a few other examples, a few other projects that we have going in the lab. And the interesting thing is that these projects began separately by different people and without us having seen a connection a priori. And what that means is that basically the model with the same equations, the same variables, but the variables mean different things. So temperature will no longer be temperature, but something else. It turns out that the same model with the same mathematical structure applies to different settings. So for example, we find that the mouse embryonic stem cells grown on a plate quorum sense, and that they help each other basically survive and grow during replication, only if you start out with enough number of cells in a population. And it turns out that the same model, but slightly tweaked with different parameter values, is able to explain, for instance, how to enable yeast cells to survive and grow at temperatures between near freezing temperatures. And I'm not showing the data here, but it also turns out that the same kind of model explains how gene expression restarts from dormancy once you wake them up. And so, the way I view this is as an experimentalist is my big hope is that there will be a model for different phenomena, and in the case of my lab, these will be phenomena related to dormancy or cells kind of nearly stop growing, but then you can revive them. And there might be a model for each of these different phenomena, but maybe they share the same kind of features in the model. And my big sort of hope, I'm probably not the person who's going to actually do this, but that my hope is that we don't end up with like a zoo of different models that describe, let's say, a life and death transitions, but that there will be some common theme that underlies these models, and that maybe there will be sort of underlying theoretical framework like the one that Pankaj was talking about earlier. And sort of, I'm not very eloquent here, but for me, what I see as a big challenge is that the models that we build to explain the experimental findings use a language that is very different from the language that we're used to from say, statistical mechanics or some of these existing frameworks from physics. So for example, you would not use energy and entropy to describe any of the experiments that I've shown you, although in principle those exist, but they're not practical metrics to describe the phenomena that we observe in the lab. And the models actually use things like concentration of molecule or phenomenological terms like production rate or secretion rate of something. And I think for me that the big challenge is to see how we can take models that use these different metrics that you can measure in a lab, and somehow have those come out of theoretical frameworks that use very different metrics than the one that we use in the lab. So that's it for me. Thank you.
Thanks very much, Yoon. We already have some questions coming in. So the first is from James Fitzgerald, and James, you can jump on here if you want to participate in this back and forth. But James was wondering how you think about abstractions as related to the sort of true-false dichotomy that you presented at the beginning of the talk. And he gives the example that the assumption that an object's mass is located at its center is wrong, but it's a useful abstraction. So how do you think about those abstractions in the context of your work?
Yeah, so physical laws are approximations, right? Newton's law is an approximation that's still practical, and so I think in the case, to be concrete, in the case of yeast cells growing at high temperature, the picture that we have, the growth rate going down as a function of temperature is true if you walk across the phase diagram along a narrow band of initial cell density. But it turns out that so that picture is still true, of course, but if you now vary the initial cell density, as you increase the temperature, growth rate can go up as the temperature goes up. I guess, yeah, so maybe true or false is not, and so I was very careful in the beginning to say, true or approximately true in some cases.
And a second question from Janne, given that you're seeing the same mathematical structure, are you learning something about cellular physiology that's more general than the specific examples that you're looking at?
A short answer is not yet. The long answer is that what we are really able to explore here, thanks to a model actually, which quantifies the phenomenon, is really what happens at the edge. So sort of at the cusp of being able to grow as a population. If you think of a population as one living system, basically it's replicating and it's growing. And then if you change one of the parameters, temperature or cell density, or some ligand that's secreted just below some amount, then the population will die, right? It will go extinct. And there's a feature that that seems to be common for the systems that we're looking at in yeast and embryonic stem cells, just at the cusp, which is the amount of time you have to wait for, let's say, a population or gene expression to resume, growth, or gene expression to resume. And so there's some kind of scaling type of thing there that that seems common to these different systems, but we're exploring that further.
Great. I think we should move on now to Christina. Thanks very much. And there are more comments in the chat, so you might want to take a look at those. And our next speaker is Christina Huyshen, who will be the final speaker of this session, and she's joining us from Stanford.
Hi there. Sorry, 1 second while I share. Okay, does that look all right?
Yes, looks great.
Great, thank you. Okay. Thanks, Anne. I'm Christina Huyshen. I'm a postdoc in Alex Dunn's group at Stanford. Big thanks to the organizers. I'm really excited to be here and to share some results and some reflections from my work on how distinct self-organized actin patterns explain diverse parasite gliding modes. Okay, so one of the 4DCP goals, of course, is to bridge scales and connect molecules to physiology, so the function of organisms, which have cool behaviors like scurrying around on the run from predators. But in this short story, we'll use a unicellular organism to take a shortcut, connecting actin and myosin to the scurrying around of the eukaryotic parasite, Toxoplasma gondii. So one of my favorite things about today's story, actually, is that we'll be using flocking theory, originally inspired by the collective motion of birds over here on the eukaryotic tree of life, to understand actin organization in Toxoplasma parasite, way over here, highlighting how math can unify across scales and systems. So Toxoplasma is a protist, closely related to malaria parasite. It sits around quietly in the brains of 1/3 of the human population and causes disease in the immunocompromised. And to move through its hosts, Toxoplasma cells glide on substrates without changing shape. And during gliding, toxo cells show off this whole collection of distinct movements. So there's helical gliding, like you see in the top left movie here. There's circular gliding. There's twirling. And there's also back and forth gliding movements, like you're seeing in the bottom right. We know from beautiful work from many groups that gliding is powered by a thin layer of actin and myosin right at the cell surface, so sandwiched between this orange plasma membrane and an inner membranous scaffold here in gold. And this brings us to the puzzle that launched this theory experiment loop, which is, how does this thin actin myosin layer drive all these different gliding movements? So how is it organized to generate coherent traction force and move the cell? The dominant hypothesis out there was that this cage of gray microtubules directed all the myosins, oriented them to transport all actin rearward. And when that actin would catch on a substrate coupled adhesion, it stays stationary, and instead myosin motors would walk the guts of the parasite forward, move the parasite forward. But this model isn't consistent with that back and forth motion that we saw, right? And indeed, when we performed single molecule imaging of actin in live Toxoplasma parasites, we were able to track directional actin transport events at myosin speeds, but they were not always uniformly rearward. So they weren't consistent with that idea. Then an extrinsic player, the microtubules, is telling the actomyosin network how to polarize and how to organize. And in the absence of an external cue, we hypothesized instead that cell scale actin patterns emerge from local interactions by self-organization, as occurs in other analogous actomyosin systems. Okay, so now we want to understand, right, what cell scale actin organization could emerge from local interactions between motors and filaments. And to do this, we used flocking theory to develop a model of Toxoplasma actin self-organization. In other words, equations that describe the time evolution of a filament density field and a filament velocity field. So for starters, we consider a regime in which actin filaments are stable. So the continuity equation here ensures conservation of filaments. And then our filament velocity field evolves according to a series of local rules, which I'll walk you through one by one. So first, filaments are transported by myosin motors with their minus ends leading. They move at a preferred speed, the speed that myosins walk, which we know from experiments. Because of cross-linking proteins and steric effects, filaments like to align with their neighbors. And then there's a pressure-like term that prevents unphysical or unrealistic densities. And finally, we have a new term relative to kind of more standard flocking theories, which punishes filament curvature. So filaments prefer to align in a direction, sorry, of least curvature. That's this last term here. Okay. Now it's one thing to solve these equations on a planar surface, but we wanted to predict actin self-organization on the real Toxoplasma geometry, right? So we actually derived a parameterization-free surface formulation of our governing equations that can handle arbitrary curved surfaces. This was actually quite technical and very fun. We then obtained 3D reconstructions of the rigid stereotyped Toxoplasma cell shape using soft X-ray tomography. And we used these to generate surface meshes on which we solved our self-organization equations using the finite element method, generating 4D predictions for the time evolution of the actin filament density and velocity. So let me tell you a little more about these results or these predictions. So when we started with an initial condition of disorganized filaments and then let them evolve according to the two equations I just showed you, so this is a stable filament regime, the model predicted the emergence of these patches of high actin density, which will show up in a second in reddish colors, that recirculate up and down the cell in this kind of cyclosis of actin indefinitely. And what's really fun is that in experiments where we stabilized actin with an actin binding drug, we were able to see bundles of actin filaments recirculating up and down the cell, which hopefully you can make out in the movie on the right. Okay, but in normal cells, we actually know that Toxoplasma actin filaments are quite short, they depolymerize or turn over quite quickly. And we know that filament polymerization is favored specifically by proteins at the anterior end of the parasite up here. So we incorporated into our model this anterior filament polymerization and filament depolymerization everywhere. And when we do this, we're able to access sort of a new range of solution space. So our model can predict instead these largely unidirectional rearward actin flow patterns. This movie is actually still playing, but the actin velocity field has reached a steady state that looks like this. So actin is continuously flowing rearward in this cell. Okay. And another thing that's really exciting is that these predictions led us to a hypothesis, which is that these distinct self-organized F-actin states, the cyclosis, the unidirectional flow tuned by actin turnover, give rise to the distinct gliding types you saw in those opening movies. So for example, helical gliding, we know from experiments, initiates when the anterior left side of the parasite, this region here, is in contact with the substrate. So we can look there in our predicted unidirectional actin pattern, and then for a rough traction force, that's this red arrow, which is in the opposite direction as actin velocity. This red traction force propels the cell forward and the point of contact changes, and so on. And what's fun is that we see that this pattern can explain the rigid body motion observed during helical gliding, as well as during circular gliding, which initiates when the cell is on its right side, and during twirling, where torque generated by the vortical pattern at the cell posterior can explain that observed cell rotation, which occurs when the cell posterior is in contact. And then at lower rates of depolymerization, the model was instead predicting, if you remember, this back and forth cyclosis of actin. And we think that that back and forth recirculation of actin can explain the back and forth gliding behaviors. Okay, so in summary, we started off this story with a puzzle, which led to some experiments, which led us to use continuum theory and coarse graining to bridge scales. More imaging entered the story here to provide 3D geometry for our modeling. And I'm actually really excited about how this approach lets us handle non-spherical cows and leverage imaging tools and data sets, which of course are strengths of Genelia's. We then used FEM to solve our model on a complex geometry and make 4D predictions, which suggested new experiments. And the last thing I'd like to highlight is that as someone with a biology background, I kind of want to share how empowered I feel by now knowing how to use FEM to solve custom PDs on weird geometries. I kind of feel like a biologist who has learned how to fish, as they say. And I think there's an opportunity to engage more biologists like me by making these types of tools just a little bit more accessible. And then finally, big thanks to the Dunn Lab for their support, and especially to my mentor, Alex Dunn, and to Rob Phillips. The theory work presented today was done in close collaboration with him. Thanks also to Liav Segov-Zarko and John Boothroyd for their toxoplasma expertise and support. And thank you to you all for listening.
Great, thank you, Christina. The first question is from Daniel, and Daniel, you can chime in if you want to clarify anything I'm about to say, but he asked if actin is stabilized, where is energy coming from to make the wavy dynamics? Is it always dominated by the myosin?
Yeah, the short answer to that is yes. So actually the kind of picture to have here is maybe if you're familiar with traditional in vitro gliding assays where myosin motors are stuck on glass or some surface and then they're scooting actin motors around. In this case, we kind of have myosin motors stuck on the inner surface, the inner membrane, scooting actin molecules, actin filaments through this chamber. So the wavy up and down recirculation of actin that you see arises purely because of those stationary myosin motors moving actin around, and then actin filaments interacting as well with neighboring actin filaments.
The next question comes from Navesh Wadhwa, who asks, what biochemical inputs to the self-organizing system does a cell tune to generate different gliding types?
Yeah, I'm glad that you brought this up. I wonder if actually I can.
And I'll maybe add in Yanni's question here, which asks how the cell might be controlling switching between these different types of motion, assuming switching happens in response to external cues.
Perfect.
But I don't want to answer those together or separately.
Yeah, I'll try to do them together, and maybe people can chime in if I missed anything. But So one thing that's fun is in the model, we can explore kind of this phase space where we tune actin polymerization at the conoid. If you still remember my equations, this was a term we called C, and actin depolymerization or actin turnover. And we can see how this shifts our system between this recirculating cyclosis and unidirectional movement. And one thing that I'm extremely excited about is in the literature, there are many of these published observations of shifts in the prevalence of different gliding modes. So one of the biggest trends is that people would do perturbations to actin regulatory proteins or to actin itself. Like here's an example where actin depolymerizing factor was knocked out. So now filaments would have been destabilized by it, but now they're more stable. And then they see much more of this back and forth gliding motility. Or actin mutations were made in actin subdomains that stabilized filament formation. We would also expect that to kind of shift in this direction on our map here. And in experiments, more back and forth motility was similarly observed in that case. And there's kind of a host of additional observations, some listed here, where there were these kind of puzzling shifts in motility type that we now think we understand based on the way that the experiments tuned these same knobs. I think a final part to Yanni's question was about when and how cells would want to tune these things. And there, I am not sure I have as much intelligent to say. Certainly cells will activate, so they'll go from a sort of disorganized, I think, unproductive mode to these more unidirectional modes when they sense that they're external to a host cell and they need to get to a new host cell and invade. That's kind of, there's some biochemical signaling involved in activating. One of the outputs there, the field definitely thinks is to stabilize actin. So I guess that's one point of contact.
Great, and I think this is now a perfect point to transition to our discussion session with all of the speakers from both of these sessions. And thank you again, Christina.
Thank you.
Janine, okay for us to just go ahead and get started?
Yeah, go for it.
Okay, great. So maybe I will kick things off. So all of the talks today touched on various aspects of this iterative theory experiment loop. These loops, as we all know, are hard to establish, they're hard to maintain and keep fresh. So we're curious, what do you think is the most important ingredient for maintaining this back and forth? And conversely, what is the biggest potential failure mode for these loops? We'd love to hear from any of the speakers. Yeah, go ahead, Guadalupe.
Yeah, no, I mean, I think it's kind of related what the question you're asking, is how to keep the conversation going. And also, I mean, Aubrey mentioned this. I mean, I think we have a problem with communication. So how to communicate between the different and kind of fields and keep, the research going and doing new experiments. And one of the things that, new models and new projects. And one of the things that I was thinking at the level of an institution is very important are seminars, right? It's the place where, you know, a whole institute get together. But when you have different, you know, you have more theoretical groups and more experimental ones, again, sometimes the issue of the communication come up. So one of the things that I was thinking is that maybe to have, you know, seminar for all and seminars more specific could be a way to, you know, to make the communication between the different groups more efficient, or, you know, theoretician giving talk for everybody. So you need to be, you know, skip details and maybe give general views. And so to get more the, you know, everyone on the same page, and again, with the possibility to start new collaborations and new projects. Again, my experience working in interdisciplinary institution, I think this has been a problem that we face, because you need to have also, you know, more specific discussions, and very important. And again, thinking about a theoretical group, you know, you need to have those discussions. So maybe to separate and having different seminars for all more specific could be a way.
If I can sort of put maybe Christina on the spot, just because she brought it up in her talk, and that is, you know, the way she's closing this loop is she went ahead and taught herself finite element methods, and you know, things that normally, at least most of my cell biology friends will not do, and you know, will go and seek out, you know, someone who will do that for them. And I was just curious, you know, to what extent do you think, you know, what's gained, what's lost, you know, is sort of going forward, you know, should we, you know, is that, you think, an important thing to that we should be developing? I mean, you know, this is kind of returning to the, I don't know, maybe my history is going to be wrong, but kind of 19th, 18th century physics, where, you know, if you did the experiments, you did the theory, and there was, you know, some, you know, good stuff that happened, and then splitting those apart, also other good stuff happened. And anyway, just thought I'd love to hear your thoughts on that, since you plunged into the whole FEM business.
Yeah, it's an interesting topic. I mean, I think there's something gained always in taking some steps yourself towards the direction that you're engaging with. Like, maybe I took more steps in that direction that's usual, but I also had the really good luck of having people who were experts to talk to. So I think there was still that element of, you know, I was working with and getting advice from maybe true theorists. But I think it did help a lot that I was trying to learn some of the language myself, and maybe that improved the overcame the kind of communication barrier that we're talking about. And that's partly, I think, why I'm so excited about. So in my particular case, I ended up using a common commercial finite element method software. I just used it in kind of a custom way. And I think that kind of step is actually doable for many biologists. It's just that there's nothing, there's no material, there's no manual, there's no one who kind of like walks you through those initial steps. But I think if there were, it would be doable. And I think there's a lot gained in, I'm trying to say, kind of the mental shift that comes from once you've tried something yourself and you've put your hands on it, you understand its value, you kind of understand better its motivation, where the theorist perspective comes from. So I don't know, I'll stop rambling, but I think there is a lot of value in individuals themselves kind of like trying to stretch a little bit more broadly, so that they can understand the language, but also kind of the motivations and value the types of questions that their collaborators are interested in.
It looks like Daniel's raised his hand to maybe join in this discussion, and then Hannah as well.
I wanted to pick up on Anne's question about where things break down. And I think one of the things which is important is the sociology. And you know, a lot of times there's still a problem with the question of appreciation of the role of theory by the experimentalists. Right? If theorists give a talk about something with a collaborator, there's a lot of experiments in it, the experimentalists give it, you sometimes get the impression that that's not, that the theory had a very little role, even in cases where the theory might have actually started the project, right? Might have got the whole project going. So I think that that's is still unfortunately a problem. I think Christina's point about the learning and doing, I think that's one of the things which is, even if it's just doing a little project and never really going further that direction, it can play a very big role, because one also starts to then appreciate that there are difficulties and obstacles and things with the theory, and it's not something you just plug it into a black box. The other part, I think, on breaking it is something which I think is a very important role of theorists, but again, I think one rather underappreciated, is the oftentimes when it's presented with some data, and then you're supposed to think about these data and analyze it. But in many ways, on the sort of big picture, often the most useful response can be, well, this data is nice, but we can't really make progress in trying to understand things without a whole lot of other data, or a completely different kind of data, and then need some back and forth about what's possible. And I think there it can break down with the experimentalist, reasonably enough, going to someone else who's going to analyze the data and be more efficient and get things out, rather than asking to go back to square one. But I do think there's a lot of sociological problems, and that is something I think Janelia can help me with the structure and so on. But it's very relevant for the management and on hiring people and evaluating them.
Hannah, did you want to chime in on that as well, or a different related topic?
So I do want to chime in on that, but take the conversation in a little bit of a different direction. I feel that I think we need to answer this question prospectively, not retrospectively, in the following way. So some thread that I saw throughout the talks today, and I actually think it's gonna go through the whole workshop, is the fact that we can collect now a lot of data, right? There's all of this data out there, and it's just exponential growth in data. So in the way that theory in the past, or computation, helped with deriving, equations and equations of motions and stuff, is that somebody had a little bit of data and then worked for many, many years to put it into equations, right? And it was a manual process, ad hoc process, based on intuition, based on working through, going from, through the multi-scale, nature of the problem. But it was a manual process. Now the question is, with all of these data that we have, is that the process by which we're going to derive equations of motion? Or is there a more systematic, automated way to do it, given that we have a lot more data? And I don't have the answer to that, but I think it might be that the interaction and the dialogue between theory, computation, and experiments has to be thought about in a different framework now that we have all of these data. And the breakpoints might be different, and so on. So what is this interplay in the era of big data and big, and high throughput biology?
This touches on something that Michael Reiser brought up separately in terms of the role that theory might have played in the past when data was much sparser, and what role it should be playing now, given that we can collect so much. And how do we think about developing theoretical frameworks differently?
Right, and how do the theoretical frameworks emerge from small sparse data versus large data, right? Large amounts of data.
Ben, did you want to comment or have
yes.
chime in on that?
Yes, I'd like to, what Hannah said, very much what I was hoping to talk tomorrow. So let me just give some thoughts right now. I think exactly, I think we need an AI-aided computational platform. I'll call meta machine learning, or just AI with human in the loop. And the scale is very different from the small data and theory. So we really have to take advantage of the computing power for fishing out like possible hypotheses. I'll call interval machine learning, and with human in the loop, and with a very quality controlled process, so that we have the process which we delegate to the machines is quality controlled, and also that part also with human in the loop. And in a particular area in machine learning right now, it's called distillation. It's kind of moving in that direction. So I will present something. We distillate wavelet can feel like more interpretable features than from deep learning, which is pretty much a black box. So I think that's exactly what has to happen, really, to match the scale. I also want to echo the discussion on sociology. I think the incentive structure has to change. Winner takes all just doesn't work. And we also need the matching of integration of mathematical models through the collaborative computing platform. Hopefully we have also AI, our statistics genie, to ask the questions. But also I think part of the model has to be also algorithmic. I don't think we can figure out all the mathematical symbols. Mathematics is not, it's very limiting at certain things. To also allow codes to be part of the description of also theory. Yeah.
Boris, you raised your hand around the same time to have something to chime in on this.
Well, I think I'm going to comment on this question of big data, which clearly is looming large. And so it seems to me that it's true that we can collect a lot more data at high throughput, and there's a lot of data, but we have to remember that the phase space of biology is larger still. We are collecting, so trying to populate infinite dimensional space with data. So ultimately, there's plenty of room in this infinite dimensional space for where we should target our data collection efforts. And so ultimately, this question of theory is a little bit the question of theory as industry or theory as art. And in this infinite dimensional space, there's clearly room for both.
And so maybe toggling back to something that Pankaj raised earlier, do we need sort of new theoretical frameworks in this area of 4D cellular physiology to be able to prioritize new data? How might we look at that moving forward, or take a forward-looking view of theory? Boris, I didn't know if you specifically had any thoughts on that before I toggle over to Genevieve. Okay, Genevieve, did you want to chime in?
So I have lots of things to say, but I noticed there's not lots of time left. So I just wanted to quickly plug that Kristen Branson actually put a lot of these questions in the Slack chat, because I think that a lot of the role of machine learning in terms of bridging the gap between big data and theory is a real topic that we're going to touch on tomorrow. So I just wanted to refer people there too, because she did a nice job of outlining questions, and I don't want to hog the next 5 minutes, so I'm going to put some stuff there.
Awesome, thank you. No, great to continue these discussions in Slack as well. Ned.
Yeah, I wanted to touch on what I think Daniel referred to as the sociology side of theory and experiment. And so, my group and I have been benefited over the years from many long-term collaborations with experimentalists. But I should point out that none of these came about because I carefully went out and decided what was interesting and who I should collaborate with. They all came from some random collision, hearing a talk, a 5-minute talk by a postdoc. I mean, it was, these things were critical then to the long-term health of my research program. And so one could think about how to, facilitate this kind of semi-random interaction. I thought the suggestion of having seminars that would be mutual interest, but just anything that, coffee and cookies, short talks, various opportunities to hear about what your theorists are doing or what experimentalists are doing, to see opportunities is important. Now, of course, not every collision formed a bond. So the need is for many, many collisions in order to form a few of those strong long-term specific bonds.
Theorist experimentalist speed dating events, maybe.
We actually literally do that during some of our junior
That's great.
That's great.
Maybe
so what are the challenges? Oh, go ahead, Vivek.
I was just going to say one thing about how in neuroscience, I think the theory has played a huge role over many decades. But the kind of role it's playing now is made possible by just the advances in tool development. So the fact that you can manipulate systems, I mean, this is something Pankaj brought up in the chat way earlier this morning, but the fact that you can manipulate the system and monitor it at a certain resolution dictates the types of implementation level things you can get to in theoretical ideas or models. And to me, what's been amazing is the developments there experimentally. So obviously theory has been useful at different levels all the way through, but at least in terms of tightening the loop and in terms of making things faster in terms of the loop, we've been able to test ideas quickly because of these advances, just bedrock of tools that, and reagents and data that's been available. So that's where certainly for us, the speeding up has happened, as you know.
But maybe building on that, so looking ahead, to what extent do we need new theoretical frameworks to enable these loops to happen faster and tighter as tools develop alongside those? Is it enough, say, in this new area that there are tools being developed, or do we need to start to formulate or expand existing theories to be able to make the same iterative progress that we've been able to make in neuroscience?
I mean, I'm just going to chime in quickly. I think it also goes to what Daniel was saying earlier. It seems to me we definitely need new math, new theory. The question is, how do we get there, and how do we put in incentives in place that people are, I mean, they're not going to happen by random chance. You need to have people out there who are going to spend who are going to be excited enough and have have the support to spend their careers on thinking about these things. And so I think that's kind of an important part of the puzzle. And I don't have any answers, but I do think at some point we have this fantastic data. There's so many cool puzzles, like Christina showed one, Ned showed one. These puzzles that we can solve with models and theory, but then we'd like to get to the point where we're also deriving kind of lessons that span many models. Something that Hyun brought up, that are of the kind that Vivek and Pankaj raised, of the kind of like Hopfield models and things and attractors and these kind of very broad ideas that can maybe not be successful in explaining a specific system, but are actually incredibly successful as putting a whole field in a certain frame of mind that leads discoveries in a certain direction. So how do we get those I guess Feynman referred to them as psychological states produced so that fields can move in productive new directions? I mean, that's a incredibly, I think, important question. And I don't think AI and these wonderful technologies that exist out there to sort of help us reduce the space that Boris talked about are going to be, maybe they'll be helpful, but I don't think they'll get us all the way there. That's just my kind of sense of it.
I know there are several hands that are raised. I think that for time's sake, we should break here and continue this discussion in the Slack. Thank you to everyone who spoke and participated in the discussion. And let's continue this on Slack.
Thanks, everyone. It was a great first session. Let's take a quick break and resume in about 7 minutes at 1:30 Eastern.